Hello guys, welcome to Sagacious Learning, and in this video we will learn about the chemical composition of the cell wall. So cell wall was discovered by Robert Hooke, and he was an English biologist, and one day he was examining a thin slice of a cork under a crude microscope, and on examination he found some perforations on that very piece of cork. So those perforations were named cell by him, and the thick membrane that was bounding those perforations was called a cell wall. So cell wall is actually metabolically very much active, and it is capable of growth, but it is considered to be a non-living structure. So if we look at the chemical composition, then it consists of fibrils, and these fibrils are actually embedded in the amorphous matrix. And as you can see in the picture also, it has been shown that how amorphous matrix is present there, and macrofibrils are arranged in this amorphous matrix. So fibrils are actually complex carbohydrates such as cellulose, and apart from cellulose there is also some presence of hemicellulose, pectin substances, lignin, cutin, suberin, chitin, and moreover calcium and magnesium are also present in the form of carbonates and silicates. So cellulose is actually a polysaccharide, and it consists of 3000 to 10,000 glucose molecules that are interlinked by 1,4-glucosidic linkages, and cellulose is thus present in the form of a long chain of glucose molecules, and these chains of glucose molecules are lying parallel to each other, so they form a bundle in this way. So when there is a bundle that consists about 100 such chains that are lying parallel, they form a mesoll, and when 20 mesolls are stacked parallel, they are said to form a microfibril, and when 250 microfibrils are lying parallel, they are known to form a macrofibril. So macrofibril is a very large bundle, and hemicellulose is present between this macrofibril. Hemicellulose is actually a monosaccharide, and it contains xylose, mannose, arabinose, galactose. Then we have the pectin substances in the cell wall that consist of glucuronic acid and galacturonic acid. Then we have lignin. Lignins are the coniferyl alcohols, and these coniferyl alcohols are actually colorless and crystalline organic substances. Then we have the presence of cutin. Cutins are composed of fatty acids. Then chitin is there. Chitin is actually a polymer of glucosamine. So if we look at the composition, then cellulose is present there in the concentration of 10 to 15%. Then we have cellulose from 5 to 15, and majority of cell wall is actually water that is present in the concentration of 60%. So you can see the majority of cell wall is water, and proteins are there from 1 to 2%, lipids from 0.5 to 3%. Then we have the pectic substances from 2 to 8%, and suberin and cutin, they are also present there. So they are impermeable to the water, and therefore they are relatively in higher amount in those cells that are directly exposed to the sunlight, because of the fact that they are impermeable to the water, and in this way they prevent a loss of water that otherwise would have been transpired. Okay, so lignin is there. Lignin provide rigidity and strength to the cell, because it reduces the water content of the cell wall matrix. And in spite of the presence of lignin, cell wall actually allows some of the water and dissolved substances to pass through as such. Then and this composition of a cell wall is not exactly the same in all the cells. It may show little variation. It depends upon cell. This is the end of the video. Thanks for watching.